In this recording, we're going to learn a bit about blood typing and transfusions. Now, under normal conditions, our bodies make all of the blood we need. Within that blood, of course, we have the liquid portion, the plasma, which helps to, along with the solutes, or the things that are in the blood, helps to maintain blood pressure and blood volume. There are cells within the blood that you should have seen now. They're suspended within the plasma, and those help to transport things throughout the body, as well as some of them serve as a part of the immune system. So under normal conditions, we've got all the blood we need, but we do know that as humans, things don't always go as normal. Things are not always normal condition. So perhaps you've had an injury or a surgery or a disease in which you're losing a lot of blood. And in those cases, you might need to have a transfusion. So it's important that we know a bit about blood typing and how that determines what blood is put into a body in those cases where blood volume may be low. So that process is called transfusion, and that's from a Middle English word that literally translates to mean transfer by pouring. The first recorded attempt of a blood transfusion was in the 15th century. There was a pope that had fallen into a coma, and his physician called in 3 young boys and actually drained blood from those young boys and was going to give them money as payment for that. And so they took the blood from the young guys and gave it to the Pope by mouth. And of course the Pope died, as well as all 3 of the young men that had donated their blood. So we've come a long way since then. We know a lot about blood typing, and we of course use a much more sophisticated method of transfusions now. So the delivery method, as well as the discovery of blood types, has led to a great increase in our knowledge. So let's talk a little bit about blood groups and blood typing. Erythrocytes, commonly called red blood cells, and all other body cells have on their membranes a genetically determined set of antigens. So these are little glycoprotein. Let me go ahead and get the pen before we get much further and get it up and running. I may want to highlight some words for you or something, so let's get this going. And you know I'm going to use red, blood red, right? Okay, so we've got these glycoprotein surface markers that are known as antigens that are on the surface of cells, and the ones we're of course talking about are the erythrocytes, or the red blood cells. Now these are genetically determined, so your parents have determined what markers, what antigens will be on those cells. But the purpose for those is to allow the body to identify those cells as belonging to yourself, so your immune system doesn't attack these. So what antigen is something that causes your body to create an antibody, and an antibody is a component of the immune system that will bind to an antigen. So you create an antibody, it binds to an antigen, and then marks that particular cell or that antigen for destruction. So we're born with all these antigens. Now, the immune system doesn't recognize foreign antigens, so if we have a bacterial infection or a viral infection, or perhaps we get a transfusion of blood that is not compatible with our own, then the immune system recognizes that and creates antibodies. All right, you're going to get more of that in the immune system, so that's as far as we'll go right now. Okay, so when antibodies that are created by the immune system bind these antigens, they form a complex. And that complex, in turn, is going to bind more and more and more and more, and you end up with this big clump. Okay, so when you have this big clumping wad of antigens and antibodies, and now remember those antigens are attached to the blood cells, then you've got this big clump of blood cells. You've got erythrocytes with antibodies attached all over them. Well, this clumping process is known as agglutination. All right, agglutination is simply that conglomeration, or that wad of cells and antibodies. And because it causes agglutination to occur, antigens are also known as agglutinogens. Agglutinogens. Break that down. It causes agglutination to occur, gen beginning. So it's cells that cause the onset of agglutination to occur. Now, because we do have these markers on the surface of the erythrocytes, or the red blood cells, we can use those to classify blood into groups, and that's where we're headed with this. All right, so let's move on to the next slide here. Now, humans have a whole lot of different antigens. There are 30 varieties of naturally occurring red blood cell antigens, but there are hundreds more that can be specific to certain families. So, like I said, you get these from your parents, so they're genetically determined. Now, most of these cause very little agglutination to occur. So if you get a transfusion and some of these minor antigens are present on the blood cells, then very little is going to happen as far as agglutination. But the ABO and RH, which we're headed to in just a few minutes, the ABO and RH antigens cause a very, very vigorous transfusion reaction when they're put into a body that does not recognize that as their own. So when this happens, of course, this can lead to a very, as it says, very vigorous reaction and ultimately could lead to death. Okay, this happens, of course, because of a mistake. So let's say a person with type A blood is given a transfusion of type B blood, then you have a transfusion reaction occur. Once again, more coming up. So let's move on and look at these blood cells now. This, I really like this figure because it shows you using different shapes and different colors to identify the antigens as well as the antibodies that are created. So the ABO blood type has two antigens, A and B, that are found on the surface of the erythrocytes, and two types of antibodies, anti-A and anti-B. All right, anti, remember, means against. So if you have type A blood, as we have here in this first picture, okay, type A blood is type A, and I guess I probably should get a different color here. Let's go with maybe green. All right, type A blood has type A antigens. But now created in the plasma, we also have type B antibodies. So it's an antibody against B, or antibody anti-B. All right, now you can see this and this are not going to bind together. Okay, so these particular antibodies look at these antigens and they don't fit. Okay, it's like putting a square peg into a round hole. That antibody won't fit that antigen, so it's not going to bind. And of course we wouldn't want antigens against our own red blood cells, right? All right, let's move over to the next one. We've got type B blood. All right, in type B blood, we've got B antigens found on the erythrocytes, and we've got type A antibodies, anti-A antibodies. Now look at these two. This and this are not going to bind together. So the antibodies, type A, will not bind to the antigens on the type B cells. Now that's very important because it's the binding, remember, of antibodies and antigens that causes agglutination to occur. As we continue, this is going to become more and more and more important as we continue to discuss this. Now, you've probably also heard of someone having type AB blood. Oops, I forgot. I can't go low down on the slide like that. You've heard of someone having type AB blood. Well, back up again. Get the pen going again. I'm sorry, guys. I know this is frustrating for you. It's frustrating for us as well when this happens. So all we can do is apologize and ask you to bear with us. Let me get the pen going. Come on, baby. All right, let's try again. Now here we have type AB blood. You can see there are antigens A and antigen B on these erythrocytes. Okay, so it's known as AB blood because the erythrocytes have A and B antigens. But what happened with the antibodies? If you look out here in the plasma, there are no antibodies out there. Well, of course there's not, because if you had A antibodies, it would bind to your A antigen. If you had B antibodies, it would bind to your B antigen. All right, and agglutination would occur. So type AB blood has both types of antigens, but no antibodies against A or B. All right, now let's move over here to type O. Type O blood, if you look at the membrane around this erythrocyte, you see there are no antigens present. All right, so there are some of the other minor antigens. Remember, we talked about how everybody has those, but there are none of the A, B antigens present on the type O blood cell. Okay, so if we have no antigens present in the cell, then we can create both types of antibodies. Now, these antibodies are going to occur in the body shortly after birth. We build antibodies spontaneously to the type of antigens that we don't possess. So if you have type O blood, you have no antigens on your cell surface, on the cell surface of your erythrocytes, so you can create both A and B antibodies because there are no antigens to bind to. All right, so this is very important. It's very important that you understand that your blood type is determined from your antigens, and then in your plasma you have the opposite type of antibodies. All right, let's move on and talk a little bit about what happens when a transfusion reaction occurs. Now, if you look at this, we're talking about antigen type A. Remember, we've seen this already. So type A blood has what type of antigen? Has A antigens, right. Well, what type of antibodies do we find in the plasma of type A? Yeah, I know you can read. We've got type B antibodies. Now, let's say this person had an injury and was sent to the emergency room, and no one cross-matched or typed the blood of this patient with the blood that they were going to transfuse into this patient. So what if they gave this patient type B blood? Now, here are your antibodies that are found in type B, right? Okay, type B blood has B antigens, but type A antibodies. So let's say we infused or transfused this type B blood into this type A patient. Here's what's going to happen. Now, I want you to look at this. These A antibodies are binding to these A antigens. You've got another binding here, A to A, another binding here, another one here, another one here, and here, and here, and you can see how this just goes on and on and on. So you're creating this huge complex of antibody antigens, and that's causing agglutination to occur. Now, when agglutination occurs, of course, this blood is not going to travel through the bloodstream as it should. So it's going to cause a transfusion reaction. Now, let's look at these photos here on the right side and talk a little bit more about this, because this is what you're going to see in lab when we do our blood typing with our simulated blood kit. Let's start with an easy one down here, like the drawing we've been looking at. So let's start with type A blood. Now remember, type A contains agglutinogen or antigen. Let's use the easy word, right? Antigen A, okay? Now, all of these in this row have anti-A serum, or serum containing anti-A antibodies. All in this row is serum containing anti-B antibodies. So here we have, remember, this has anti-A antibodies, this has anti-B antibodies. Now we put our type A blood onto this slide. All right, so here we put our type A blood. Here we put our type A blood into the serum. Now remember, this serum contains anti-A antibodies, AB, I'll just abbreviate for antibodies. This one has B antibodies. Now look what happens. What's going on here? All right, if this serum had A antibodies, what type of blood did that serum come from? I know you said type B, right? So B blood has anti-A serum. So here's type B blood serum and type A blood serum. All right, so look what happens in these pictures. All right, we've got type A blood. We put in type B serum, and we have agglutination occur. Look at what's happened to those erythrocytes. They're all wadded up. Now, the purpose of erythrocytes is to carry things throughout the body. Well, this is not going to be very efficient at carrying anything throughout the body. Imagine if you're walking down a hallway. Now, you're an erythrocyte. That hallway is a blood vessel. Now, if you're walking down through there and there's nothing interfering with you, you can go on and go do whatever it is you're headed to do. But suppose you run into a crowd of people and everybody's holding each other, arms are linked together. All of a sudden, you can't get through that hallway. Well, that's rather simplistic, but it's the same process that's going on here. When you have these conglomerations or these agglutination events occurring, then the erythrocytes can't go on and do their job. So look at each of these. Here's your type B blood. Remember, we have B antigens. Now here's our type B serum. Nothing happens. We wouldn't expect it to, right? But then look over here at our type A serum, which has anti-B antibodies. Once again, agglutination occurs. Well, let's look at type AB. Remember, type AB contains both A and B antigens, right? So in this particular serum from the type B blood and from the type A blood, you're going to have antibodies binding antigens because you have both types of antigens. So whichever antibody you have in that particular serum is going to bind with that particular antigen. Now let's move down here to the very last one, the type O blood. Type O blood, remember, contains no antigens. So regardless what antibodies are in your serum, there's nothing for it to bind to. So you have no agglutination occurring. All right, let me peek right quick and see if there was anything else I wanted to tell you about. Okay, so what happens when agglutination occurs? Well, it should be pretty obvious that the blood flow is going to be restricted, especially through the small blood vessels. So you're not having blood get out to the tissues. Well, why does blood go out there to begin with? You've probably heard this already, right? Blood goes out to the tissues to carry oxygen and to carry nutrients and to bring back waste. So the transport function, that's a vitally important function. Also, maintenance of heat and all the other functions you've heard about. Well, if the blood's not going out there, all that's not happening. So you have reduced transport, reduced distribution, reduced maintenance of homeostasis. Now, there's also a serious side effect of this as well, and that is that these erythrocytes that are agglutinated will eventually begin to lyse. So they undergo hemolysis. Where in the world can I write this word? Let's change colors just so it will stand out a bit more. Hemolysis. You've seen lysis before. Remember, it means to split, and hemo means blood. So this is the splitting or the breaking down of those erythrocytes. Well, when this happens, it releases all of the hemoglobin molecules into the bloodstream. Now, when this happens in excess, you've got all these free hemoglobin molecules that are flooding the kidney tubules. And this can pretty quickly lead to cell death, and if something's not done about it pretty quickly, also can lead to kidney failure, which can lead to death of the patient or death of the person receiving the improper transfusion. So the treatment for these transfusion reactions typically is centered around diluting the blood by giving a lot of fluid and also giving diuretics to help to wash all of that excess fluid out. So dilute it down, wash it out quickly, so all of that excess hemoglobin is going to be washed out through the urinary output. All right, let's see where are we headed next here. I think next is just our summary table. Okay, now let's look at this table, and it has the same information that I've already given you, as well as the population or the percentage of the population that has each of these blood types broken down by ethnicity. So you can see that In the Caucasian population, 45% have type O. In the African American population, 49%. The Asian population, 40%. And the Native American, 79%. So, what's the most common? Exactly, type O. And what do you see is the least common blood type? 4, 4, 5, and less than 1. That would be AB. So AB is the least common blood type, and type O is the most common. This also shows you the same thing that we've already looked at. Here are your blood types. Okay? Here's an illustration with your differently shaped antigens, the types of antibodies that are found in the serum, and then it also tells you what type of blood can be used in a transfusion with this type of blood recipient. So let's look at this for just a few minutes. Notice this column, blood that can be received. All right, if you've got type A blood, what type of antibodies do you have? None, right? You got type A and type B antigens, but you've got type none antibodies. So you can get type A, type B, type AB, and type O blood. So this is what's known as the universal recipient. All right, let's move on to the next one. Type B blood, remember you've got type B antigens, anti-A antibodies. So there's our anti-A antibodies. So what type of blood can you receive? Well, of course you can receive B because that's a perfect match. But also type O. Now think about that for just a minute. Why could you receive type O blood if you're type B? Or type A. Let's go ahead and talk about that one as well. Because you see here type A and B both can receive type O. Think about that for just a minute. I see the wheels turning. All right, let's look at this. Now remember, type O has no antigens on the red blood cells or on the erythrocytes. So the antibodies, are not going to be able to bind to those cells. Okay, so you've got type A antibodies here, type B antibodies here, but on these type O erythrocytes, there's nothing for them to bind to. So they can indeed go ahead and take type O as well. All right, so that covers type A and type B. Let's go ahead down here and talk about type O blood. Now remember, there are no antigens on the erythrocytes, but there are type A and type B antibodies. So with no antigens for the donor, or no antigens on the donor cells, then this is the universal donor blood type because there are no antigens on the red blood cells for the recipient's antibodies to attach to. All right, I hope that makes sense to you. Take some time and really think about this. This is some really important information. Now let's move on and talk about one more blood group or one more antigen, and that is the RH factor. Now, RH was first isolated, and I guess I better go away from the red and get a different color again. We'll go with blue this time. RH was first isolated from rhesus monkeys, and that's where your RH came from. It was later discovered, of course, to be a component of human blood as well. So these are also antigens that are located on the red blood cells. So if you have these RH antigens on your red blood cells, then you're RH positive. RH negative, bet you can figure that one out, huh? RH negative, no RH type D. Type D is the most common of several different RH antigens, so it's usually the type D that is considered when you're looking at RH positive or RH negative. Now, about 85% of the population are RH positive. All right, so about 85% RH positive. That's quite a large percentage there. So now remember I said earlier that the ABO blood types or the ABO antibodies, I'm sorry, were formed shortly after birth, spontaneously formed against the antigens that we don't have, right? So if we have type A antigens, we form type B antibodies. You remember that. It was just a few minutes ago. Okay, but now the Rh antibodies don't form spontaneously. It's like the immune system has to be triggered to form these. So let's say an Rh negative individual receives Rh positive blood. Well, the immune system is going to recognize that Rh factor, that Rh antigen on those erythrocytes, and it's going to form those Rh antibodies. Now, that's usually not a problem, or almost never a problem during the first transfusion, unless it's just a massive, massive quantity of blood and it goes on for a long period or an extended period of time. And the reason for that is because it takes the immune system some time to build those antibodies. They have to recognize those antigens, and then there are certain cells in the body that are going to become antigens, and you'll hear more about that in the immune system. But it takes some time for those Rh antibodies to form. So the first exposure typically has no effect. But now let's say that this happens again, and this Rh negative person receives Rh positive blood. Then you have the typical transfusion reaction as you saw earlier. Now, one of the most common ways for this to happen is Rh incompatibility with an Rh negative mother and an Rh positive father. There's dad, there's mom, and lo and behold, they've created a baby. Guess you know what they've been doing, huh? All right, you better know after the last unit. All right, so what happens here? Let's say that this baby, and I apologize, I know this writing is blurry, so hopefully I can talk you through this. We've got an Rh positive father, Rh negative mother, and they conceive an Rh positive baby. So baby is Rh positive, okay? Now remember, mom is Rh negative. Well, guess what? It's no big deal for a while. The only time that this becomes a problem is, one, if the mother has an abortion, two, if the mother has a spontaneous abortion, also known as a miscarriage, or three, after delivery, when there may be some bleeding that occurs when the placenta breaks away from the uterine wall. Now that can cause some spillage of that fetal Rh positive blood into mom's bloodstream. Now, you've already seen what happens on the last slide. We talked about what happens when mom or when anybody gets exposure to Rh positive blood cells if they're Rh negative. Well, what happens? They start building up antibodies. Okay, so mom delivers this baby, and baby is fine, but now remember, mom's blood has been exposed to, or mom's immune system has been exposed to that Rh positive blood now, so she's carrying Rh antibodies. All right, no problem. Now we got these little antibodies, and antibodies are Y-shaped. Well, those antibodies are going to be against those Rh positive erythrocytes, right? Well, next time mom gets pregnant, she had better hope that the baby is Rh negative. Because if the baby is Rh positive, her immune system has already created antibodies that are going to bind with the red blood cells of this baby, if that baby is Rh positive. And remember what happens when antigens and antibodies bind. Agglutination occurs, and suddenly this baby's body cells are starved because the agglutination doesn't allow for the transport of blood. You also can have jaundice occur as the bilirubin builds up. I know you've probably not heard that word yet. Maybe you have. I'm not sure what was covered in the clip before this, because I'm recording this one before the other one. Anyway, bilirubin is a pigment that is a component of the breakdown of erythrocytes, and that's what causes a buildup of bilirubin causes jaundice. Now, worse than that, it can, of course, cause the developing fetus to die because of severe agglutination. So what can we do about this? Bet you're ahead of me already. To prevent this from happening, mom typically will take a shot of RhoGAM. Heard of that before? RhoGAM is typically given before or very, very shortly after birth. Now, this, of course, needs to happen here, not over here. All right, we need to have that RhoGAM shot right before or right after this birth so that we don't have this buildup of antibodies. What RhoGAM is a serum that contains Rh antibodies. So those Rh antibodies are going to bind these Rh positive cells and go ahead and mark them for agglutination while there's just a few in the bloodstream. Go ahead and mark them, get rid of them, get them destroyed, so that mom's immune system never has time to recognize them and therefore never build up any antibodies against those. Pretty cool, huh? Okay, the last slide I want to show you is just another summary slide. I thought this might be helpful to you in lab when you're doing the blood typing exercise, because it shows you in a very simplistic manner. Here's your blood type. Here are your antigens that are present. Here's your serum. Now remember, type A antigens or type A antibodies are found in B blood. Type B antibodies are found in A blood. So it shows you with a positive or negative whether a glutination or a transfusion reaction would occur if you had a mixture of those two types. Okay, so as we wrap up this particular chapter, you should have a pretty good understanding of blood, its components, all the cells that you find within it, the functions, and hopefully a few fun facts thrown in there as well. So, with all of that said, we're going to finish up the chapter on blood and move into the heart and the rest of the cardiovascular system. So you guys be studying. We'll see you soon.